Okay, well, here we go. And if I'm glistening a little bit, that's because it's hot out there. And hopefully, by tomorrow, fingers crossed, it's not going to be hot for at least some time. So, what I'm going to do is I'm going to go over this preprint paper. So keep in mind it has not been peer reviewed yet, and it came out on June 3rd. And so the title is Naturally Occurring SARS-CoV-2 Gene Deletions Close to the Spike S1/S2 Cleavage Site in the Viral Quasi-Species of COVID-19 Patients. So going to unpack a few things there. So remember, the spike protein is the protein that is basically the spikes, quite literally, they're the spikes of the coronavirus. So they're proteins that project out of the virus's membrane and they interact, at least in the case of SARS-CoV-2, they interact with the ACE2 receptor, which is how the virus gets stuck to a cell, and then after that sticking, that is when the virus is able to undergo some sort of proteolytic cleavage, which then lets the virus material get injected into the cell. Okay, so these spikes play an important role in virus sticking to the receptors. Anyways, there are multiple cleavage sites or multiple proteolytic cleavage sites on the spike protein. One of those is between the S1 and the S2 subunit. So many of you might have heard of the furin cleavage site or the polybasic cleavage site. So this junction between the S1 subunit and the S2 subunit is where that polybasic furin cleavage site is located at. And in a closely related coronaviruses, there is no polybasic cleavage site. It's just a monobasic cleavage site. However, that is also cleavable by certain proteases. But what ends up happening is this can happen before or after the virus actually is interacting with the cell. But this spot gets cleaved in order to facilitate in the virus getting into the cell. So sometimes it can happen while the virus is being produced, and sometimes it happens after the virus has attached to the cell. Regardless, there are, in a subset of SARS-CoV-2 viruses, and remember, viruses mutate very frequently, in a subset of those viruses, there are some with a mutation or gene deletions close to this cleavage site. And what this ends up doing, what this ends up doing is it produces a lot of proteins. The viruses with this mutation will produce proteins that do not have the entirety of the S2 subunit. And the reason why is that it ends up encoding the stop codon. So the stop codon, there are multiple stop codons, but one of them, or sorry, three of them, and what they do is they tell the translation machinery, or the ribosome, the thing that produces proteins, that produces these spikes, it tells that to stop making more, or stop polymerizing more amino acids. And so when that happens, the spike protein ends up being incomplete. And this happens in a subset of the viruses that are within one person. So they call this a viral quasi-species because it's not actually a different species of the virus. However, it is very dissimilar. So it's almost as if it's a different species, but not quite. Okay, so, and keep in mind, I also want to mention that this S2 subunit seems to be very important in allowing the virus to enter the cell. So if this S2 subunit of the spike protein isn't being produced at all, then we could assume that the virus that uptakes these S1 subunits. Let me backtrack a little bit. Because I believe, if I remember correctly, the membrane-spanning portions of the spike protein are in the S2 subunit, if I'm remembering this correctly. And yeah, I am, because it's at the very end of the S2 subunit, where you have a lot of the there's a lot of thiol groups or a lot of amino acids with sulfhydryl groups on them, and those are acylated to allow the spike protein to. I take that back again. I know that that's part of the fusion peptide, but I don't know if I've forgotten whether or not the membrane spanning portions are like which subunit they're a part of. I believe they're in subunit 2, and I believe the beginning portion of subunit 1 might also be in the membrane. Yeah, that's definitely true. There is some portion of the S1 subunit that is in the membrane. So, regardless, sorry for rambling. I will review the structure of the spike protein a little bit before my next video. Regardless, here they're saying that some of the viruses in a single individual can be mutation or mutants, and those mutants have incomplete spike proteins. So that's the general idea. So let's go to the abstract. That way we can get a broad picture of what's happening. So the SARS-CoV-2 spike protein, also known as the viral mediator for binding and entry into the host cell, has sparked great interest as a target for vaccine development and treatments with neutralizing antibodies. And the reason why we tend to focus our vaccine development and our drug development on these spike proteins is because they are the portion of the cell or portion of the virus that is exposed to the environment or to the plasma. And because of that, that makes it an easy target for antibodies and the immune system. Okay. Anyways, initial data suggests that the virus has low mutation rates, but its large genome could facilitate recombination, insertions, and deletions, as has been described in other coronaviruses. So remember, coronaviruses are generally very large viruses genome-wise or genetic-wise. They have a lot of information that encodes their entire structure. And because their genome is so large, that gives it a lot more opportunities to have crossover events with other coronaviruses. So the bigger the viral genome, the more opportunities that genome could cross over with a different virus, which would lend to, one wouldn't say a higher mutation rate, but a higher variability rate in the phenotype of the virus. And then also insertions and deletions, which is those, they're called indels, and those mutations happen whenever the viral replication machinery slips a little bit. So that type of mutation, at least on a per virus basis, can be pretty significant, especially if you're dealing with a really large genome. Continuing. And here we deep sequenced the complete SARS-CoV-2 spike gene from 18 patients with mild and 8 with severe COVID-19. And we found that the virus accumulates deletions upstream and very close to the S1/S2 cleavage site. Okay, so frequently we're seeing mutations right before this cleavage site. So what can this tell us? This can tell us that these mutations can be somewhat beneficial, at least in a subset of the virus. They can be beneficial for the replication or the duplication of the virus, at least within a singular person. This doesn't say anything about transmission. It means of an individual person, you can see these types of mutations. And continuing, generating a frame shift with appearance of a stop codon. So a frame shift mutation would be if you add or remove a single or 2 nucleotides, and then that, I don't know how much detail I should go into, but that basically would scramble the amino acids that are being produced by those by that genetic code. So it's almost as if you removed. I would say, think of it like this. Let's say you have a sentence, but you don't have. You remove all the spaces, and so you just can, and you can um, only read the letters, but there are no spaces. And let's say you go through a mutation, a frame shift mutation, uh, it would be very difficult for you to read the sentence because how to phrase this? It'd be difficult. So let's say there are no spaces. So the way that you read is you read 3 letters at a time and there's no spaces. So every word has 3 letters. Okay? Every word has 3 letters. So the problem that happens when you have a frame shift mutation is that if you introduce another letter or remove another letter, then every single word is going to be shifted by 1 or 2 words, or 1 or 2 letters. And so every single word ends up being scrambled with the word either before or after it. And this is what happens to viruses and this is what happens to viruses very frequently. So what happens is there is a specific word that tells the ribosome to stop translating and stop making proteins. And normally, for the spike protein, this stop codon, this stop word, would be at the very end of the protein, after the protein has been completely synthesized. However, when you have these indel mutations, it makes it much more likely to have the stop word randomly appear within the spike protein. And when that happens, you create incomplete proteins, if that makes sense. So if we continue, these deletions were found in a small percentage of the viral quasi-species, so only 2.2% of the viruses are undergoing these mutations. And in samples from all of the mild and only half of the severe COVID-19 patients. So every mild case had a detectable level of these quasi-species that have this incomplete spike protein. Okay, they all have viruses with the complete spike proteins, but the mild cases, for every mild case, there was always a portion of those viruses that had incomplete spikes. With the severe COVID patients, only half of the people had detectable levels of this quasi-species. Okay. Anyways, our results suggest that the virus may generate free S1 subunit proteins, and this is released into circulation. So why would a virus want to do that? Why would a virus want to produce a useless protein? Okay, so we propose that natural selection has favored a don't burn the house down the house strategy, in which free S1 protein may compete with viral particles for the ACE2 receptor. And when this happens, thus reducing the severity of the infection and tissue damage without losing transmission capabilities. Okay, so I can see, so what's happening, what they're proposing is happening, is that the virus is making these dummy proteins, and they're releasing them. And they're presuming that these dummy proteins are making the disease much less severe, or it's making the disease, what would you call it? They're not getting as severe pathology from this. So their proposal is some of the viruses will mutate into these dummy viruses, or these dummy proteins will be synthesized, and then those dummy proteins will interact with the ACE2 receptor in your body, which could theoretically interfere or prevent the normal virus, the unmutated viruses, from infecting the cells. And why would they want to do this? What would be the rationale for a virus to employ this strategy? Well, if the viruses don't want to kill you, let's say they want you to have a mild case so that you end up thinking you're all right, and you end up going out into society and you start spreading more virus particles around because you have mild symptoms rather than severe symptoms. That could be a strategy that the virus is employing. So what I want to do is I'll go through the background, and I will then go through the results, because I don't think going through all of the materials and methods will really matter that much. If you want to read it for yourself, I will leave a reference or a citation down in the description below. But anyways, let's go to the background. So RNA viruses, let me drink. Here we go. So RNA viruses replicate using their own RNA-dependent RNA polymerase. This isn't always true. There are some RNA viruses that do not have an RNA-dependent RNA polymerase. There are some retroviruses that, such as HIV, so they are RNA viruses. But rather than having an RNA-dependent RNA polymerase, they end up having a what is it? Reverse transcriptase, I believe, is the name of the enzyme that they use. But what they basically do is they will have an enzyme that will read the RNA and turn it into DNA. And then the DNA can exist in the cell, and then when it wants to end up creating the new viruses, your own body has your own transcriptases that are transcriptases that turn that DNA back into RNA, or I should say copy that DNA into the complementary RNA. So rather than you could argue that retroviruses have an RNA-dependent DNA polymerase because it depends on the RNA and then it produces a DNA polymer. So this isn't exactly true, but good enough. Anyways, so they are called, in the viruses that do have the RDRPs or do have the RNA-dependent RNA polymerases, this RDRP lacks proofreading mechanisms. I might have to come back on that later. There might be some proofreading mechanisms with the RDRPs. However, let's keep on going. So these people say that the RDRP lacks proofreading mechanisms and is prone to mutate at high rates. So about 1 out of every 1,000 to about 1 out of every 100,000 substitutions per replication cycle will be a mutant, according to these RDRPs. Okay, so if you have a genome of 100,000 nucleotide bases, and remember the nucleotides are the letters of DNA or RNA. If you have 100,000 of them and your mutation rate is 1 out of 1,000 substitutions per nucleotide per replication cycle, that means every time you replicate those 100,000 polymer of 100,000 nucleotides, every time you duplicate it, you will probably have about 100 mutations each time around. So if you think about the virus, the SARS-CoV-2 virus, there is probably, with its, 30,000? Yeah, 30,000 base pairs. So with 30,000 base pairs, we could estimate that every time it gets duplicated, you could have probably up to 30 mutations every time it's replicated. Okay. Now, that doesn't mean that you will get 30 mutations every time it's replicated, but you can get up to that amount on average. But that doesn't mean that doesn't mean that you are going to see those viruses when you sample people. And the reason why is there's evolutionary pressure. So perhaps there's a lot of evolutionary pressure to not mutate, right? And so if you have all that pressure to not mutate, then all of the mutants that have a lot of mutations, they might end up dying, whereas the viruses with very few mutations end up surviving and duplicating some more, right? So don't view this as, oh, viruses are going to have this many mutations every time they replicate. That's not exactly how it works, because there is selective pressure either for or against certain mutations. Regardless, you should take this as there is a lot of opportunity to mutate. There's a lot of opportunity for viruses to explore the different spaces, the different genetic spaces, in order to find a more, what would the word be? A more virulent strain, or a strain with more vitality, if you will. Even though viruses aren't alive, we can view them as having vitality to them. Anyways, with these mutations, this is lending the virus a quasi-species structure. So let us look this up just to make sure that I have the right definition in my brain, because when I think of quasi-species, I think of like fake species, like but that might not be. So the term quasi-species describes the type of population structure in which collections of closely related genomes are subjected to a continuous process of genetic variation, competition, and selection. Usually, the distribution of mutants or variants is centered on one or several master sequences. Okay, so that is something, that is a good distinction to put in. So a quasi-species, you could view it as population genetics. So think of how all humans are one species. We are one species. We are one kind. But we do have a very small amount of genetic variation within our genomes. And the distribution, the population density, if you will, of those different genes can vary a little bit, and over time they can change a little bit, and there can be selective pressure to change those a little bit. And you could argue that if you view mankind as a superspecies, that we all contribute to altering our environment in some way, then there might be different phenotypes that might arise based on the distribution of genes among that population. So with viruses, there is if you get infected with a virus, the virus within you is invariably, no pun intended, it's invariably going to have a bunch of different variants within your body. And so what pathology you receive, or how the virus influences your metabolism, that is going to be dependent on not just one virus's genetic makeup, but it's going to be dependent on the distribution of the genetic makeup of all of the viruses that exist in your body. Okay, so, and we could argue that some viruses might play different roles. So a virus might decide that it doesn't want to duplicate, but it might want to produce proteins that make it easier or harder for the other viruses to duplicate, right? So you can also view this from the lens of some sperm cells. Some sperm cells will have, not all sperm cells are going to fertilize the egg, but some sperm cells will have specified functions that help one of its own kind end up reaching the egg to help it compete with another mate. So it's not just, so for instance, with garter snakes, some of the sperm and semen will end up forming a plug, which prevents the garter snake from being inseminated again. And that would be a way to, for a sperm cell to pass its genetic information, not through itself, but through one of its very close relatives by sacrificing itself to form this plug. Anyways, I'm going on tangents, and I wanted this to be a short video. Continuing, previous studies with severe acute respiratory syndrome coronavirus, or SARS-CoV, and mouse hepatitis viruses, which I believe is another coronavirus, have reported moderate mutation rates of, oh, these are small mutation rates, so 9 times 10 to the negative 7 and 2 times 10 to the negative 6 substitutions per nucleotide per cycle, or per duplication, which is very low. So normal RNA-dependent RNA polymerases are normally 1 out of every 1,000 to 1 out of every 10 or 100,000, but with SARS, it's 1 out of every, oh shoot, what would that be? About 10 to the negative 6, so about 1 out of every million. Yeah, about 1 out of every million, and 1 out of every 2 million. Do I have that right? One out of, oh gosh, I hate math. So this is about 10 to the negative 6. So yeah, 1 out of every million for this one, and this is 1 out of every, I would say, 400,000. I think it's 1 over that. Regardless, very, 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 on a per nucleotide basis, very low mutation rates for SARS and mouse hepatitis virus, which I believe is another coronavirus. So, and this is, as I said, below the expected range for RNA viruses. But keep in mind, they are long genomes. So long genome has more opportunities for these mutations, even though it is low on a per nucleotide basis. Anyways, this is consistent with the role for non-structural proteins, NSP 14, in RNA proofreading or repair functions because of its 3' to 5' exonuclease activity. So this is why I was a little confused. They said earlier that RDRPs do not have proofreading mechanisms, and I think that they are right in that the enzyme itself does not have a proofreading mechanism. However, there is a non-structural protein, or an NSP 14, so it's probably called NSP 14, or it's probably open reading frame something, but regardless, it is not a part of the virus's structure. It's this accessory protein that the virus produces, and this accessory protein has a proofreading mechanism, at least in these coronaviruses presented. And it's called an 3' to 5' exonuclease, in that it has the ability to split up or break up what would you call it? A nucleopolymer. So things like DNA or RNA, but in this case it's an RNA. Anyways, nonetheless, the large size of the coronavirus RNA genome increases the probability that deletions will be generated and recombinant events will take place, which could facilitate adaptation to new host environments, as occurs with jumping between species. So a recombination event is when genetic material is existing in the solution, and recombination is when you can have multiple strands of genetic material, and then somehow they are able to cross over. So this could be done through, say, an exonuclease, which breaks some of them, and then they'll end up just coming together, and then a polymerase can come by and basically sew the genetic material back together. I do not know the exact mechanism by which RNA viruses do that, but that is the general gist. Somehow the genetic material can become excised and then interact with another strand and then get fused together that way, or at least spliced together that way. Anyways, one naturally occurring deletion on 29 nucleotides in the open reading frame, or ORF8, of SARS-CoV after human-to-human transmission was found to be associated with attenuation of replication, so lowering of replication. So in the original SARS-CoV, there was a deletion of 29 nucleotides. So 29 letters of the genetic material were deleted, and this genetic material was associated with the protein that was encoded that is called open reading frame, or ORF8. Okay, they'll just call it ORF8. And when this accessory protein, this is not a structural protein, when this accessory protein had this deletion, human-to-human transmission, one naturally occurring deletion of 29, yeah, so after this deletion, the virus became a lot safer to humans. So somehow the deletion of ORF8, or at least the mutation of it, by removing a big portion of it, this ended up making SARS a lot less able to replicate in the human body. So they're arguing that inserts and deletions can play a pivotal role in the pathogenicity of viruses. Anyways, the low mutation rate and the high human-to-human transmissibility, R naught of 2.2, and absence of human pre-existing immunity against SARS-CoV-2 could explain its rapid spread through the human population with very high sequence identity. So there are multiple factors in play, which is why the SARS-CoV-2 hasn't, at least on a nucleotide basis, it hasn't mutated that much. So it has a low mutation rate, right? As probably due to some, it's probably due to some proofreading mechanism by a different protein of the virus. So that's the low mutation rate. It has high human-to-human transmissibility. So the same virus from one person can infect a lot of people. And that would be one way to have the virus be less variable. They're using an R naught that I don't quite agree with, because I believe it is much higher normally. But this is the R naught that they have, that there is a lot of some consensus about between 2 and 3, but it's probably between 3 and 6 based on the death data. And the absence of human pre-existing immunity. So immunity causes selective pressure. So if you are immune, or at least if you are partially immune to a disease, your immune system is applying evolutionary pressure against that virus from duplicating as easily as it wants to in the body. But since humankind has absolutely no immunity against SARS-CoV-2, that means that the virus, when it gets into your body, it knows that it's going to duplicate very readily. It can duplicate without any issues at all, because you have absolutely zero immunity to it. So if you could imagine, let's say you have a virus that you do have some immunity to, and you get infected with it. Well, your immunity is going to kill a lot of the viruses that look similar to the virus that you were immune to. But that's going to leave a bunch of dissimilar viruses with unique genetics circulating within your body. And those unique viruses are going to, since they have selective pressure to be different from the original disease that you had earlier, that means that evolution is pushed forward, or mutations are brought to the forefront. They can survive a little bit better than the original disease. So that's one way that you can provide selective pressure towards mutation. So, and they are arguing that these elements combined is one of the reasons why there is high sequence identity among uh, all of the SARS-CoV-2 coronaviruses that have been genetically analyzed. The high pathogenicity of the virus, the severity of COVID-19, and the lack of an effective antiviral treatment or vaccine has pushed the scientific community worldwide to develop, in record time, a solution for this pandemic. I mean, we need to find a solution before we can say it's in record time, but I get the sentiment. Among the SARS-CoV-2 structural proteins, this includes the spike protein, or the S, the envelope protein, or the E, and the membrane, constituting the viral coat, and the nucleocapsid, N protein, that packages the viral genome. So nucleocapsid proteins find the genetic material and they encompass the genetic material, but around that nucleocapsid, there is still a membrane, denoted M, and that membrane has the spikes and the envelope proteins associated with it. The spikes are what allow the virus to interact with the cell, and the envelope proteins are structural proteins that help make the membrane be in the right shape. Anyways, the S-glycoprotein is the most promising as a therapeutic and vaccine treatment. So since the spike protein is a protein that is actually pointed out at the environment, that is where we decide to do a lot of our research into therapeutics. The S protein is encoded by the S gene, and following trimerization, so the spike proteins are trimers, so 3 proteins will come together to make the final spike. It composes the spike of the characteristic viral particle crown, so they call this the corona, hence coronavirus. The S protein is essential for the SARS-CoV-2 to infect a host cell, and by recognizing and binding to the human cell receptor, the ACE2 receptor, and possibly with lower affinity to other receptors. I would like to see this. So there is the possibility for some receptors, I think receptors on a subset of the immune cells. That is something. And then also when it comes to antibody-dependent enhancement, that would be utilizing the Fc receptors of some cells as well. And possibly with lower affinity to other receptors, such as the CD209L. So this is a different receptor, and I can look it up just to make sure that we have the right. So this was for the original SARS. So this is not the one I was talking about earlier, but CD. Maybe, let's say in COVID-19, let's see. So yeah, I'm not seeing anything here that I wish to bring up. Just know that this is another receptor that might interact with the spike protein. So that's another thing that we have to consider is these spike proteins, or these proteins, that viruses express in order to get into different cells. They can be, what would the word be? Promiscuous. They can be promiscuous in what receptors they bind with. So if you can imagine, you have some viruses that interact with one receptor, but then then let's say it infects another animal, and this other animal, has that same ACE2 receptor, but maybe it has a different receptor that actually binds just as well. Well, then that virus can use that other receptor and then perhaps start mutating to become better and better at using that different receptor. And that would be one way that a spike protein can interact with one receptor normally, but then over time mutate to work towards a different receptor. And that could be in a virus's best interest, because a virus just wants to get its genetic material into the next generation, right? So you can accomplish this by being promiscuous, at least to a small degree, with your spike proteins. Anyways, the S gene has 3,822 nucleotides with 1,278 amino acids. It has 5 essential domains: the receptor binding domain, or the RBD, and this is in the S1 subunit. It also has O-linked glycan residues flanking a polybasic S1, S2 cleavage site. So this S1 subunit and the S2 subunit cleavage site. In SARS-CoV-2, this is a polybasic furin cleavage site. And then around it, there is an O-linked glycan, or there's a sugar that links to an oxygen on the protein. But I don't remember reading this. In fact, I thought that they were N-linked for the most part, but apparently there's an O-linked one as well. And then there's a fusion peptide, which is near the end of the S2 subunit, and it has heptad repeats, HR1 and HR2, and I haven't read too much about those. And it has a transmembrane domain. Anyways, the S1 receptor binding domain includes 6 amino acid positions that show high affinity for the human ACE2 receptor, which is widely distributed, but mainly present in alveolar type 2 cells of the lungs. So ACE2 receptors are all over your body, but it's mainly one of the main parts that is at least exposed to getting a virus, at least initially, is the alveolar tissue, or the alveolar type 2 cells of the lungs. Once the virus is attached to the host cell receptor, cleavage occurs between the subunits S1 and S2. This might be a little bit, some people might argue that it's already cleaved. It's pre-cleaved in the viral processing before the virus has even been released into the environment. So as the virus is being synthesized, some people have seen that there is already cleavage that has happened between the S1 and the S2 subunit. However, I don't know if we have done a study on the virus. I don't know if we've done microscopy yet on SARS-CoV-2 that has been able to see whether or not the spikes were complete or not. Continuing, and subunit 2 drives the viral and cellular membranes to fuse. So the fusion machinery is in the S2 subunit. Thus, S1 recognizes and binds to the human cell receptor ACE2, whereas S2 directly facilitates entry into the host cell. So both functions are crucial for infection, and therein lies the interest of spike as a target for the development of vaccines and antiviral agents. So because of the importance of the S protein, we carried out a deep sequencing study of the spike gene in the upper respiratory tract samples from 18 patients with mild or severe SARS-CoV-2 disease. And of particular note, hotspot deletion sites were found in minority mutants located upstream and very close of the S1, S2, and the S2 prime cleavage site. So it's not just the S1, S2, it's also the S2 prime. And this S2 prime is within the S2 subunit, and it is another proteolytic cleavage site that might facilitate the virus in entering the cell. Anyways, this is suggesting that these genomes code for a truncated or an incomplete S protein. The variants were significantly more prevalent in patients with mild than those with severe disease. So they're saying here that the people who were lucky enough to have these mutants in them tended to be to have more mild disease. So, thus, their effect on the protein could constitute a favorable regulatory mechanism emerging in the viral quasi-species to modulate the pathological effects of infection. So it could be that some of these viruses are mutating and they make the disease less severe in order to help you survive. Because remember, viruses do not want to kill their host. That is not their goal. Their goal is just to multiply. Killing their host is not their goal. Anyways, discussion is provided on the implication this observation may have in the biology of SARS-CoV-2. So, the patients and methods, just know that they collected some patients and they were categorized as either severe or mild. And then the detection of SARS-CoV-2 was utilizing in-house polymerase chain reaction, which is a way to a way to duplicate areas of genetic material, and then that can tell you whether or not you have the sequences that you're looking for. But that's just to verify whether or not the genetic material is present. And then you have the SARS-CoV-2 sequencing. So they take the respiratory specimens, and then they are able to do sequencing on it. And I won't go into too much detail because I forgot a lot of the ins and outs of the sequencing. Just know that they can do the sequencing, and then they can get a distribution of different genetic material, they can get a distribution of genomes that are found within a singular person, and then they can do some studies on that. Anyways, bioinformatics, the Indel study. So, for one, I forgot. Actually, I don't think I was ever taught this because I think this is cutting edge stuff as far as being able to sequence a lot of things at the same time and then get that distribution. I think it was cutting edge when I was in school. So, just know that they're going through these samples and they're getting a bunch of different genomes from each sample, and they're getting a bunch of genomes from the SARS-CoV-2 coronaviruses that were in their samples. So here are the results. 18 coronavirus disease patients were included in the study, and in total, 48,700,000 reads, and they were ranging from 81,000 to about 600,000 reads per amplicon. And they were analyzed from the upper respiratory tract samples. And using 13 overlapping amplicons covering the complete spike protein. Thus, we studied 3,700,000 complete spike genes with a mean of 208,000 spike genes per patient. So, trying to, let me get the precise definition of an amplicon for you. It's a piece of DNA or RNA that is the source and or product of amplification. So amplicon or replication events. So it is a sequence of DNA that they have duplicated multiple times. So, and these amplicons overlap with each other to a degree. And this is how they are able to synthesize the full genome. But with this overlapping, they can get, I would say it's sort of like a distribution of different genetic material that is all aligned, so they align it all together, and this genetic material is found within each sample. And on average, there was about 208,000 unique, how do I phrase that? It would be the equivalent of studying 208,000 unique spike proteins of the per patient. Now, that isn't necessarily, if I'm remembering this correctly, if I'm reading it correctly, that doesn't mean that they studied 208,000 complete S protein sequences. It's that they have the genetic equivalent of that much. So they might not necessarily know one complete spike protein, but they can see the variations within each area of the spike proteins, and on average is about 208,000 reads of genetic material for a complete spike protein. Regardless, results related to the amplicon positions, coverage percentage of the master sequence, gap incidence per patient and per amplicon, and premature stop codons are available in supplementary tables S1 to S10. So remember, the stop codons are important because that can tell you whether or not your spike protein was truncated. So remember, if you add, remove, or in some rare instances, a single mutation, if you do those mutations, you can every once in a while create a truncated version of the protein because you have accidentally added a stop word to that genetic sequence. Anyways, uh, but the deletions were not randomly accumulated along the S gene. Okay, so if it was truly a random process, which it probably is, the deletions themselves are probably random. You could probably find them equally distributed among uh, all the RNA or all the genetic material if there wasn't evolution at play. So evolution is going to screen out, remove, or prevent the duplication of useless mutations, whereas beneficial mutations will remain. And so deletions were not randomly accumulated, so they are accumulating over time, but they were not randomly accumulated. They instead were found in specific regions where they tended to at least be conserved. And deletions coded at delta 1 to delta 18 range from 1 to 42 nucleotides lost. In some cases, the sequence recovered the correct reading frame. In others, the frame shift caused the appearance of a premature stop codon where very close to the deletion site, whereas in still others, a new amino acid segment appeared. So, sorry, I just got a shooting pain in my arm for some reason. Probably spending too much time on the internet. So, deletions were found in all amplicons, but yeah, it's probably from having my hands up there for too long. So deletions were found in all amplicons, but they were mainly observed at frequencies less than 1. So these deletions are rare. They're not good for whatever effect they have. It is an effect that is very potent. So not too much of them, or not too many of the amplicons show these deletions. So most deletions in amplicons, NO4, NO5, NO6, and nucleotide 4, 5, 6, 10, 11, 12, and 13 were found in only 1 or 2 patients. Whereas deletions in amplicons 1, 2, 8, and 9, ranging from 2 to 16 nucleotides, were observed in 4 to 9 patients. And a deletion of 6 nucleotides in amplicon 7, this generated a stop codon, and it was present at a frequency of almost 4% of the quasi-species in samples from patient 9 and patient 17. The largest deletion, which involved 42 nucleotides, was found in nucleotide 5 of patient 9 and resulted in a loss of 14 amino acids. But the reading frame was recovered. So the reading frame being recovered means that how do I phrase that? That it might lose or gain certain amino acids, but after that loss or gain, the amino acids are the same. Anyways, a striking result was the accumulation of deletions in amplicon number 7 between nucleotides 1980 and 2035. In 14 out of 18 patients, which included 100% of patients with mild disease, and only half of those with severe disease. Okay, so a striking result was the accumulation of deletions in amplicon number 7. And this amplicon number 7 was between 1980 and 235 in that spike protein. And I believe this would be encompassing the cleavage site between the S1 and the S2 subunits. Okay, and only half of those with severe disease. In this particular hotspots, deletions of delta 2 and delta 34 were produced. Among among severe patients, P12, P13, and P16, or patient 12, 13, and 16 had no deletions in the amplicon 7. Oh, shoot. Okay, sorry. I shouldn't call these nucleotides. No, these are amplicons. So amplicon 4, 5, 6, 10, 11, and 12. So, yeah, sorry, I said nucleotide, but really I meant amplicon. And only half of those with severe disease. In this particular hotspot, deletions delta 2 to delta 34 were produced. And delta means deletion, I believe. But a deletion of 2 nucleotides to 34 nucleotides. And among the severe patients, patient 12, 13, and 16, they had no deletions in this amplicon. And patient 17 showed a deletion outside the hotspot location. Viral variants carrying these deletions were significantly more frequent in mild than severe COVID-19 patients. So this could tell you, if this is true, these people are receiving more of these deletion mutations, and apparently these deletion mutations might make the disease less severe. But remember, this is only happening in a very small percentage of the total viruses in the body. So most of the viruses in the body are functioning as normal. Anyways, among the total of 43 delta 15 deletions in amplicon number 7, a premature stop codon appeared immediately after the deletion site in 5 cases. So after this particular deletion, there was five cases of a stop codon. And the reading frame recovered after losing 4, 5, or 7 amino acids in 6 cases. However, a frame shift that changed the reading frame and caused the appearance of a premature stop codon several amino acids later was generated in most of the deletions. And in consequence, the S1, S2 cleavage site and the polybasic domain disappeared. So in a lot of these mutations, these frame shift mutations, the spike protein is being truncated and it's losing a lot of the S2 subunit and some of the S1. In 39 of the 43 amplicon 7 deletions, Tata box-like motif, forgot what, I'm going to look this up. I remember Tata boxes, but I forgot exactly what, so a Tata box, or a TATA box, is a DNA sequence that indicates where a genetic sequence can be read and decoded. It's a type of promoter sequence which specifies to the other molecules where transcription begins. So transcription is a process that produces an RNA molecule from a DNA sequence, but one would also say that since this is dealing with RNA, it's going to be different from the DNA version. So that's why they call it the Tata box-like motif, I'm guessing, and its loss. In this particular region, the deletion was characterized by a similar 3' cutting edge. An interesting result at the amino acid level was that regardless of the starting position of the deletion, in 9 of the mild patients and 2 in the severe ones, the frame shift caused by the deletion generated a new peptide motif, RLRLILLGGHBV, with the stop codon at the end. So, depending on what type of frame shift mutation you have, you can get a new peptide motif and with the stop codon at the end of it, which is truncating the spike protein. So, in 9 patients, a second, and what I will do is I will save this because I think I want to do, what is it, PepBank? So PepBank. Let's see if this peptide shows up anywhere in the PepBank. And it looks like no. Let's do a blast search. So search. So the most similar one I can find is this. So 8 out of 9 positive, so that's a lot. So arginine, um, this is so the impact of hepatitis B virus, basic core promoter, and remember, I think hepatitis B virus is another RNA virus, but I could be mistaken. Basic core promoter, mutations on T cell response to an immunodominant HBx-derived epitope. IF and gamma secreting T cells recognize previously unknown immunodominant CD4 positive T cell epitope. That is this. In 86% of patients, and this peptide bound several HLA DR molecules. So HLA is the human leukocyte something. It's basically the major histocompatibility complex. Its coding sequence overlaps the domain of the HBV genome, encompassing the basic core promoter region, taking into account the selection of viral core promoter mutants during HBV infection. We found the HBV variants with BCP mutations were present in patient sera. We further demonstrated that these viral mutant sequences activated T cells specific for the immunodominant epitope only weakly, if at all. This is the first study linking BCP mutations and HBx-specific T cell response. So conclusion, wild-type and variant peptides may represent a potential tool for monitoring the HBV-specific T cell response involved in sequence evolution during disease progression. Finally, the degenerate HLA-DR binding of this promiscuous immunodominant peptide would make it a valuable component of vaccines for protecting large and ethnically diverse patient populations. Okay, maybe. I'll look into this a little bit more, but let's finish this because we're almost done. So in 9 patients, a second deletion hotspot was found, deleting a number of nucleotides from 2 to 16 between positions 2,451 and 2,467, coinciding with the secondary S cleavage site, which is the S2' cleavage site. The hotspot was located between nucleotides 2431 and nucleotides 2454, just after exact S2' cleavage sites, which is the KPSKR. So lysine arginine is a pretty good cleavage site because it's dibasic, and in fact, a lot of proteins in your body can recognize the lysine arginine cleavage site, especially for the production of some bioactive peptide hormones. So yeah, let's go back to this and then we should be done soon. So discussion. That's a long discussion. Okay. Do I want to finish this? I think these people are basically just repeating what they said earlier. So yeah, I think rather than going through their discussion, I'm just going to leave it. You can read their discussion if you want. I'm just going to insert a few of my opinions right now. So it's getting late. I'm tired of this. So why would I can think of multiple reasons why a subset of viruses would want to mutate to make truncated versions of the spike protein. So one of the reasons I could see is that, for one, it might increase the, or I should say decrease the severity of the COVID-19. By decreasing the severity, you end up allowing the virus to exist longer in the body, presumably or probably, That's one reason. This would allow you to have a more mild case, which would allow the virus to infect more people, possibly. Another reason I could see is that they could be decoy spike proteins. So I think there's a possibility with Ebola viruses that Ebola also was able to produce, or some people argued that Ebola was producing these decoy proteins so that uh, you would make antibodies against those decoys, and then since they were decoys, it ended up increasing the survival of the virus itself, of the Ebola virus. So that's another thing. That's another thing that this virus might be employing is maybe a subset of the viruses end up being dummy viruses, and then they, for lack of a better word, they sacrifice themselves to just exist as somewhat screwed up proteins in the environment. That is true. Another thing I could see is that maybe the virus wants you to make antibodies against the S1 subunit, because perhaps there are epitopes, or perhaps there are amino acid sequences that if you make antibodies against those sequences, it ends up suppressing the immune system in some way, or at least it makes it difficult for the immune system to attack the virus in some way. That is another possibility. I don't see why it would be useful on the S1 subunit, since that is useful for the virus in general. Well, I could see it. I could see the use when it comes to antibody-dependent enhancement. So, for those of you who don't know, well, that would normally make disease worse, I would assume. Maybe there's a balancing act. Okay, so bear with me. There is a technique called antibody-dependent enhancement that some viruses can employ, which is when, if your body makes antibodies for the spike protein, or makes antibodies for the virus, and if those antibodies aren't perfect, if they are imperfect at being able to make enough antibodies, then what happens is the virus can use those antibodies to get into immune cells and then start viral replication within those immune cells. But when they at least in coronaviruses with SARS, antibody-dependent enhancement usually was studied with the antibodies against the receptor binding domain. But the receptor binding domain is usually tucked away within the spike protein, and so it's very difficult to make antibodies against that. So, if I was a virus and I wanted to convince you to make antibodies against this receptor binding domain, and maybe by doing this, well, I guess that doesn't make too much sense, because there's usually some receptor binding domains that are exposed on a subset of the virus. Or all of the viruses, I should say. But let's backtrack. What if the virus wanted you to make antibodies against this receptor binding domain, but you usually are protecting your receptor binding domain? We'll make a bunch of imperfect S1 subunits. And if you make a bunch of those imperfect S1 subunits, your body might start producing a lot of imperfect antibodies. Imperfect antibodies. That would be a very effective way. So let's say you're a virus. You don't want to kill your host. right? So you don't want to duplicate a lot. But at the same time, you want to live as long in that host as possible. So what do? If I was a virus, I would make a bunch of dummy virus particles. And these dummy particles would go out, and your immune system would act upon those dummy particles. But those dummy particles aren't exactly like what you look like, right? Because they're truncated and they're the shape is a little different and the amino acid sequence is a little bit different. So let's say these antibodies start existing in solution. What will end up happening is the antibodies will be generated. The virus will probably be a little bit suppressed, and especially if there's a lot of antibodies, the virus will definitely be suppressed. The virus will definitely go down in concentration in the body if there's a lot of these imperfect antibodies. And let's say later, eventually the antibody concentrations go down a little bit. And since they're imperfect antibodies already, then once they go down a level, the virus can then use those imperfect antibodies to undergo antibody-dependent enhancement later, and then basically cause a reinfection event to take place. Because the whole time, the antibodies were never perfect. The antibodies were never great. Would that be an effective strategy? Maybe. Would this mean that people with mild cases, they go through sort of like a sinusoid, or they will have peaks and valleys of their disease progression because the virus is never truly being killed. It's just being suppressed for a while by imperfect antibodies, and then eventually it builds up, and then your immune system then responds again, and then suppresses them later. I personally think that that would be a very effective strategy, and especially if those imperfect antibodies were also screwing up with the hormones of the host, that could be another strategy by which the virus uses to stay in circulation for far longer. I mean, we're having instances where people are they're testing for the antigen, or the viral proteins are being detected over a month after they've supposedly been cured of their infection. And the argument is that, oh, those are just like leftover remnants from when the virus was originally there. Well, especially if those remnants are activating your immune system in some way, then you're not really cured, because you still have the irritant in your body, right? Yeah, that would be a strategy. Now, granted, we could be anthropomorphizing viruses too much by saying, oh, they're definitely employing this strategy, or, oh, that's a really interesting strategy that they're employing, when really they're just randomly mutating. And the strategy is just what had the strategy is just the removal of all the unsuccessful strategies and leaving behind the successful strategies. That's basically what these viruses are doing. But I can definitely see the possibility that a subset of virus proteins are being released, imperfect and truncated in some form, and then those truncated versions are serving as some sort of decoy, which can create imperfect antibodies, which can then be used for antibody-dependent enhancement later along in the virus's life cycle. This is truly, if that was the technique being used, that is truly remarkable. Now, granted, that may or may not be what's happening. But yeah, I'll look into that. I I will do some literature searches to see if viruses actually do employ decoy proteins frequently, because that would be fascinating. I really need to pick up a book on viruses, learn more about them. Anyways, thank you for listening. I will hopefully have this video up by the end of the day as well. So hopefully everything goes as planned. And thank you for listening. So I'll see you later.